Now you're going to define what a hormone is and how it is distinct from a neurotransmitter, and then at the very end, go over some brief information about what hormones do functionally and how we study them. Hormones are chemicals that can vary in structure based on their organic building blocks. For example, some are lipid-based or based off of small fat molecules. Some are peptide-based, meaning they're based off of the small amino acids that go into proteins, and some are steroid-based. Hormones are released from endocrine glands that are ductless in nature, meaning that the chemicals go directly into the blood and produce effects on target cells by binding to specific receptors. Some effects produced by a hormone are caused by intracellular cascades involving the nucleus of the cell, transcription of genes, and translation of proteins. Other short-lived non-genomic effects occur as well, without the need for transcription or translation. Hormones coordinate the physiology and behavior of an animal by regulating, integrating, and controlling its bodily functions. Many hormones produce multiple effects. For example, luteinizing hormone causes the sex gametes to mature and stimulates mating behavior in many species. This dual function likely ensures that mating only occurs when animals have mature gametes available for fertilization. Hormones are similar to neurotransmitters in some ways, but it's also important to recognize several key distinctions. First, hormones act more broadly along wider gradients. As you will recall from biological basis of behavior, neural messages during neurotransmission must travel from neuron to neuron, involving usually a mix of electrical and chemical signaling. And this often occurs along nerve tracks, especially in the peripheral nervous system. By contrast, hormones are released into the blood where they can act on target cells throughout the entire body. Neural messages are also digital, all or nothing events that occur rapidly, like flipping on and off a standard two-position light switch. Hormone messages are analog, graded events that can occur over varying periods of time, like turning on a light using a dimmer switch, but then you decide whether you would take seconds or hours to turn it on and how high you actually want to turn it on. Hormones produce their effects by binding to receptors associated with specific target cells throughout the body. Some of these exist on the cell, but there are also receptors that can exist intracellularly. Lipid hormones, especially, can even enter the plasma membrane of the cell. However, if there's no receptor present on or within a specific cell, there will not be a hormonal effect. A hormone binding to a target receptor produces either a genomic or a non-genomic effect. A genomic effect again means that the hormone activates genes that regulate protein synthesis. A non-genomic effect means that the hormone produces more rapid effects that are not mediated through transcription and translation. Hormones are stored in and released from endocrine glands. Unlike duct glands like sweat glands or mammillary bodies that secrete their chemicals onto bodily surfaces or into an internal organ, endocrine glands are ductless. Being ductless means that endocrine glands secrete their hormone chemicals directly into the bloodstream. Endocrine glands have a sensing and signaling system that can detect feedback from target cells to regulate the duration and magnitude of their analog release events. Much like a thermostat, this function is essential for establishing homeostatic balance. We will go over several specific examples of feedback regulation later in the course. The endocrine system is also organized differently than the nervous system. although some similarities exist as well. Both are major communication systems in the body that integrate stimuli and respond to changes in the external and internal environment. Both coordinate functions of differentiated cells, tissues, and organs. However, the major anatomical distinction is that the endocrine system is discontinuous. Unlike highly interconnected and anatomically conserved central nervous system structures like the brain and spinal cord, the endocrine glands are dispersed throughout the body. For example, the pituitary and pineal glands exist in the brain, the thyroid gland exists in the throat, the adrenal glands exist on top of the kidneys, the pancreas exists behind the stomach, and the ovaries and testes exist in the pelvic region. The nervous system conducts information differently than the endocrine system. The nervous system exerts point-to-point control through nerves using electricity and sometimes a mix of chemical transmission. Much like a telephone signal, these all or none digital signals are relatively fast. In contrast, the endocrine system exerts a broad, widespread analog signal, much like a radio broadcast. Specifically, a graded volume of a hormone is secreted into the blood through ductless glands so the message can reach almost all cells throughout the body. However, the cells do not require a receiver. They do require a receiver, or in biological terms, the receptor to get the message. Without a receiver, in terms of music through the radio, or a receptor in the cell, no message will be received. A cell is a target because it has the specific receptor for the hormone. Most hormones circulate in blood, coming into contact with essentially all cells throughout the body. However, a given hormone usually affects only a limited number of cells, which are called target cells. A target cell responds to the specific hormone because it bears receptors for that hormone. So if no receptor, it doesn't matter how much hormone is present, without a receptor or a target receptor on the specific cell, you will not have an effect. So let's repeat that one more time because this is important. So if there's no receptor, it doesn't matter how much hormone is present, no receptor, no effect. Now that you have a general idea of what a hormone is and how endocrine signaling is distinct from neural signaling, let's consider the principal functions of the endocrine system. The endocrine system is involved with the maintenance of the internal environment in the body or maintaining the optimum biochemical environment. It's also involved in integrating and regulation of the growth and development of the organism. It also controls uh, the maintenance and instigation of sexual reproduction, including gametogenesis, coitus, fertilization, fetal growth, and development and nourishment of the newborn. So, in general, the endocrine system is involved in the coordination of the physiology and behavior of an animal by regulating, integrating, and controlling its bodily function. But this is hormones and behavior. We're also interested in how hormones are affecting behavior. So let's consider that as well. So, what is behavior? One way of thinking about this is that behavior is simply an output. But in the case of hormones and behavior, it's also important to consider that behavior can also be a feedback into the hormone or endocrine system that can regulate endocrine output. So, how might behavior affect hormones? Most research does not look at this, but it again is very important to consider as well as to how hormones affect behavior. They play off of each other. Behavior can and often does affect hormone levels, which in turn can influence subsequent behavior. One fun example of this involves the World Cup. Uh, one study looked at hormone levels, specifically testosterone changes of fans in a World Cup finals match between Brazil and Italy. Brazil won on penalty kicks, a very sort of nerve-wracking, frustrating finale of a soccer match. And what the researchers found was that the Brazilian fans showed an increase in testosterone when they won on these penalty kicks, whereas the Italian fans all showed a decrease in testosterone. Again, showing how behavior can impact hormones. Behavior can and often does affect hormone levels, which in turn can influence subsequent behavior. Another example here would be comes from a study where testosterone concentrations were measured in four heterosexual couples over a total of 22 evenings. On 11 evenings, saliva samples were obtained before and after sex. On the remaining 11 evenings, two samples were obtained, but there was no sex. Having sex, it turns out, caused an increase in testosterone levels in both male and female identifying partners. No changes were seen on the no sex nights. The early evening samples revealed no differences in testosterone concentrations between sex and no sex evenings, suggesting that sex itself increases testosterone more than testosterone causes sex. Alternatively, physical exercise may have been involved in this as well as an alternative interpretation. So, the study of hormone behavior relationships is organized around the idea that animals are composed of three interacting components: input systems or sensory, integrators like the brain and spinal cord, and output systems, including muscles. Here is a general schematic of how these three component parts can interact and feed into each other. Behavior can impact hormones. Hormones can impact the central nervous system, like the brain or the central processor. This can also impact sensory systems and muscles, which can all alter behavior, leading to a feedback into hormones themselves. So, how do you study whether or not a hormone can actually impact a behavior? To do this, or to satisfy the criteria to determine that a hormone is impacting behavior, you want to meet three conditions. To satisfy this causal link, you really need to meet all three of these conditions. First, you want to observe that a hormone dependent behavior should disappear if the hormone is removed or the actions of the hormone are blocked. Second, you want to observe if the behavior stops, uh, and if so, if the replacement of the hormone will restart the behavior. Third, you want to observe whether hormone concentrations are directly related or covariant. This would mean that the higher the hormone, the concentration, the greater the display of the behavior, assuming that there are a sufficient number of receptors or target receptors in place. So first, I'll have you think back about our first lecture on Berthold's roosters. Do all of these criteria fit? Another example would comes from zebra finches. A series of experiments have demonstrated that removing the testes of a male zebra finch stops it from singing. But then if you reimplant the testes or provide testosterone supplements, the singing resumes. And it also has been shown that this singing is very important for reproduction. Um, right. So this demonstrates that so again, these three conditions must be satisfied by experimental results to establish a causal link between hormones and behavior. First, you need a hormonally dependent behavior uh, to disappear if the hormone is removed or the actions of the hormone are blocked. Second, if the behavior stops, replacement of the hormone should then restart the behavior. So this would be example like if once you remove the testes from the zebra finch and they stop singing, you can then give testosterone injections and the singing will resume. And then third would be that the hormone concentration should be directly related to the behavior in question. There are several issues here, of course, involving the study of hormones and behavior, the relationship between them. This third class of evidence is very difficult to obtain because many hormones have a long latency of action and or long offset latency and are released in a pulsatile manner. But most studies or attempts to assess hormones rely on single blood draws or saliva samples. Therefore, it's oftentimes insufficient to measure changes along over a wide time course of which hormones are released, oftentimes in this pulsatile manner, to correspond or relate that to behavior. I think in the future we will probably have better technology, possibly even, in the most sci-fi terms, although I've seen these devices in development. But you might even have tattoos, for example, that can integrate with your smartphone and give you feedback or biofeedback as to what your hormone levels are. We're just not quite there yet. We're still at the level of doing single blood draws or saliva samples at one specific point in time. Another problem is that biologically effective amounts of hormones are tiny and very difficult to measure accurately. This is one reason that some of these microsensors that have been played up have not actually been very functional in practice. And additionally, unfortunately, the first two classes of evidence are very important for reliability, but hard to determine in humans. You cannot easily remove the testes from humans to see what effect that might have, for example. So most human studies tend to be limited to just this third classification system, which is in and of itself very limited by this first point.